Protein expression is the process on how living cells or organisms synthesize, modify, and regulate proteins. It also can refer to application of laboratory procedures required for protein production in protein research. It also a major laboratory tool in biochemistry, molecular biology, and protein research, as well as other scientific research fields that rely on functioning proteins. Protein expression system is the type of living organism that is used to express the target protein. It is a genetic construction which is a gene encoded by DNA intended to create a protein within or outside of a cell. It also employed in protein research as well as commercial production of enzymes and medicines. However, it can be difficult due to cloning issues, which are inefficient transformation, poor host development, inactive protein due to inaccurate folding, insoluble protein, and ineffective protein purification. Therefore, the first important step when it comes to protein expression is to choose the right expression system, for example, the E. coli expression system. So, how proteins are made? First, proteins are synthesized and regulated in response to the cell's functional requirements. Then, blueprints of proteins are encoded in DNA and decoded by highly regulated transcriptional mechanisms to generate messenger RNA, which is mRNA. The message coded by an mRNA is then translated into a protein. Transcription is the transfer of information from DNA to mRNA. Translation is the synthesis of protein based on a sequence specified by mRNA.
Hello and Assalamualaikum. My name is Muhamad Nasharudin bin Nawi, and I'm going to discuss about factor selection of protein expression system. So why we need to consider factor of before selecting our expression system, because a poor choice can lead to the inactive protein or poorly yield protein in the expression system. So before starting the recombinant protein project, we must consider a few factors. So there are a few factors for selection of protein expression system, which is time, where the time is the time spent on the expression system to yield the protein. And then mass, where the mass of the proteins, if it higher than 100 kilodalton, then mammalian or cell-free expression system is more suitable. Otherwise, if the mass of the protein is less than 100 kilodalton, then baculovirus or bacterial expression system is more suitable. And then destination of the protein, whether it is for surface or for free use of the protein. And then the need of post-translational modifications. So if the protein needs to undergo glycosylation, then a mammalian protein expression system is more suitable. And then the amount of the protein, same as the mass. If the amount is higher, then a mammalian or insect is more suitable. Otherwise, baculovirus or bacteria or yeast expression system is more suitable. And then the ease of handling the expression system. If the staff or the procedure is more suitable for mammalian, so mammalian expression system is likely to be used. Otherwise, if the easiness of handling the expression system is toward the bacterial system, then bacterial system is should be used. Thank you.
Next, we're going to talk about types of protein expression system. The first one is the bacterial expression system. The frequently used host for this system is E. coli, and the others are Bacillus subtilis, Bacillus brevis, Bacillus megatherium, or Caulobacter crescentus. Basically, bacterial expression system promotes large-scale production of recombinant protein in a short time, in which the doubling time of E. coli is 20 minutes. It also requires simple culture conditions such as media and additives, which are scalable and low cost. The E. coli BL21 and K12 and their derivative are most frequently used for routine protein expression. However, this system could cause accumulation of proteins as inclusion bodies due to overexpression of cytoplasm. It also can cause protease contamination from host protein, which lead to degradation of expressed protein and endotoxin accumulation, or LPS. The steps for bacterial expression system started with transformation of competing E. coli with the gene of interest. Next is selection of transformed E. coli by using an antibiotic and expansion of selected E. coli to a higher scale in appropriate culture media such as urea broth medium. Lastly, the isolation and purification of the protein of interest. Next is yeast expression system. The most common hosts used for this system usually Saccharomyces cerevisiae, while the others are Yarovia lipolytica and Pichia pastoris. This system can generate large quantities of recombinant eukaryotic protein. Usually, the promoter used for recombinant proteins of S. cerevisiae is Galactose inducible promoter, CAL. Besides, yeast cells are grown in well-defined media and easily adapted to fermentation, allowing for large-scale and stable protein productions. It is also capable of modifying complex proteins, but have slower growth than bacterial cells. So the steps of yeast expression system begin with the transformation of competent E. coli with the gene of interest and selection of transformed E. coli by using an antibiotic as a selection marker. Next is expansion of selected E. coli to a higher scale in appropriate culture media such as LP medium and isolation of DNA or another expression vector such as plasmid. Then, transformation into yeast and screening the transformants for integration of DNA into the yeast chromosome. Lastly, the selection and scaling up of high-expressing yeast clone in appropriate culture media and isolation of the protein of interest.
Hello and Assalamualaikum. I am Ahmad, Shafiq, Aminuddin, Musa. I'm going to talk about the insect, mammalian and cell-free expression system. Firstly, the insect cell expression system. The vector used is baculovirus and the host is fall armyworm or Spodoptera frugiperda as the host. The insect cell expression system are also known as the baculovirus expression vector system. This system uses the cell line that is SF9 and SF21 as the recombinant protein expression. The baculovirus is a lytic DNA virus that is routinely amplified in cell of the Lepidopteran insect family. It is non-infectious in vertebrate and its promoters are inactive in mammalian cell. The example for the insect cell expression system is back-to-back baculovirus expression system. In the first stage, the selection and insertion of gene of interest into the plasmid. The second is insertion of the transformed plasmid into vector. Thirdly, selection of the vector which contain the transformed plasmid. Fourth, transfection of vector to the insect cell line. And lastly, the amplification and expression of the selected protein. The mammalian expression system. The vector that is used in the mammalian expression system is the plasmid of E. coli or cell-free or the virus via transduction. The mammalian cell is used as the host of the expression system. The mammalian expression system are ideal for expressing mammalian protein that require multiple post-translational modification for proper protein function. Most of the DNA construct developed for mammalian expression use viral promoters such as SV40, CMV, and RSV for robust expression after transfection. The mammalian system can also express protein transiently and to stable cell line and can be used constitutive and inducible promoter. Example of mammalian cell-based expression system is the stable expression and the transient expression. The stable expression takes months to produce protein but can produce many proteins. The transient expression can produce proteins in a week but the protein produced are only inadequate amount. Lastly, the cell-free expression system. This system uses lysate of cell as the vector and it is an in vitro synthesis of protein using translationally competent whole cell extract. The proteins that are used in the system is assembled in vitro utilizing the purified transcription and translation machinery strands. The system is ideal for the rapid assembly of more than one protein in one reaction. It can assemble protein with tagged or modified amino acid which are helpful in the various downstream applications. It is costly and technically demanding to use. The example of cell-free expression system is the one-step Chinese hamster ovary high yield in vitro translation trial kit. This is the procedure of the kit.
Hi, my name is Nesalata and I'm going to explain the pros and cons of different recombinant expression system. As for the bacterial expression system, one of the pros of using it is it causes high expression level of the desired protein. Secondly, it is low cost and fast breeding. And thirdly, it is simple. The vector construction is very simple because the main method of using this expression system is we transfer the vector which has the inserted target DNA of the protein that we want to express into the host cell. And the cons of using the bacterial expression system is the post-translational modification processing of this expression system cannot be done because it is imperfect. And secondly, there can also be potential of endotoxin contamination as such from gram-positive bacteria. And thirdly, improper folded protein will be formed in the cytoplasm, such as the insoluble aggregates known as inclusion bodies, which then leads to the difficult purification of the desired protein. One of the pros of the yeast expression system is it is simple to culture because it has been used since 1996 in the brewing and the bread industry. Secondly, it is also capable of eukaryotic post-translational modification because it contains the yeast expression system contains the merits of both the prokaryotic and eukaryotic expression system, and it also does not cause hyperglycolization. And thirdly, it also produces high protein yield. The cons are, first of all, it is not suitable for high density culture, and it also lacking strong and strict regulation promoter because the secretion efficiency of the yeast expression system is low, because most of the target protein has molecular weight which are greater than 30 kilodaltons, which causes the secretion to be low and the secretion of the desired protein. And fermentation is required for elevated yields for this expression system. For the insect cell expression system, we mainly use bacillovirus because it is the most studied and applicable insect virus. And this insect virus, the expression of it causes high level of protein expression up to 50% of the total protein amount. Why do I say that? Because the DNA replication as is assembled inside the bark of the bacillovirus, which has great flexibility and it could accommodate large fragments of DNA insert. And secondly, it is more scalable than mammalian cells because in this protein expression system, it has limited host range and it does not infect the vertebrates without any modifications. And thirdly, it has complete biological functions such as protein correct folding and disulfide bond. As we go to the cons, the first is the exogenous protein expression is under the control of the very late viral promoter, which means the cells will begin to die due to infection. And secondly, the virus production can be very time consuming, and thirdly, it can also cause a slow growth rate. In the mammalian expression system, one of the pros of using it is it causes very good protein folding, such as the glycosylation and disulfide bonds. Secondly, stable production of cell lines can be created because the recombinant proteins expressed in the mammalian cell uses plasmid transfection and viral vector infection, in which the plasmid transfection can take weeks or even months to be done, while the viral vector infection can infect cells within just days. Thirdly, the foreign protein produced by the mammalian cells is closer to the native protein, and this causes the foreign protein has much more activity than the proteins produced by the prokaryotic expression system, such as the bacterial, the E. coli, or by the eukaryotic expression system, such as the insect cell. The cons of using this expression system is it causes low yields. And secondly, it also causes the scaling difficulties of the protein, of the desired protein. And thirdly, this expression system is complicated with complex culture conditions. And fourthly, It can also be expensive to set up and to maintain the activity done using this type of expression system. As for the cell-free expression system, one of the pros is eukaryotic cell-free lysates provides post-translational modifications such as the core glycosylation and phosphorylation, and also for the membrane protein components such as the microsomes for the membrane protein insertion. And secondly, the open system in this expression system allows addition of many components, the natural or unnatural components to the reaction, such as the labeled amino acids, detergents, and this allows us to directly manipulate the chemical environment in this expression system. And thirdly, rapid synthesis for the constructs of protein expression, because it can only take several hours to get the protein done, and we can do this in a small scale reaction up to and the volume from 50 to 100 microliter. The cons of this expression system is the production cost is high if we want to produce large quantities of the desired protein, or if the protein that is expressed shows poor yields, give us poor yields. So the production cost is going to be high. And secondly, endonucleases and exonucleases can quickly degrade the DNA of interest, which then leads to significantly reduced yields, because the plasmid, which is circular in shape, it is not affected by exonucleases, but the linear expression of the DNA of interest can be affected by the endonucleases.
Hi, Assalamualaikum. I'm Muhamad dan Hakem bin Zaki will present most common protein expression system. So, for most common protein expression system that is widely being used nowadays by biological engineering industry is bacterial expression system by using Escherichia coli or E. coli. So, why this expression system is being widely used? Because the first one, it has unparalleled fast growth kinetics in glucose salts media, and given the optimal environmental conditions, its doubling time is about 20 minutes. And also because of its easy manipulation, its easy genetic manipulation, low cost media, and also short turnaround time, which is plasmid transformation of E. coli can be performed in as little as 5 minutes. So for the next part is E. coli expression system mechanism. So this figure tells about how E. coli expression system mechanism is done in industry. So for the first step is transformation of competent E. coli with gene of interest. Next one is selection of transformed cells, test expression and expansion of selected E. coli to the higher scale, isolation of plasmid, and the last one is purification.
For conclusion, protein expression involve processes which describe how living cells or organisms synthesize, modify, and also regulate protein. Protein expression also has now become an important laboratory technology, particularly in biochemistry, in molecular biology, in protein research, in scientific fields which require functional protein in their research. Express protein products that are functional and have similar characteristics to their native counterparts also are very useful for further research, such as the determinations of three-dimensional shape of protein structure as chemical tools or probes, or as new types of drugs. The ability to express functional protein also enable researchers to study them in vitro more easily.